In this video of mediators of inflammation, we will focus on cytokines and chemokines. The references for this video are following. The proteins that are secreted by many cell types, and these proteins are mediators and they regulate the immune and inflammatory reaction, are termed as cytokines. Uh, these cytokines are secreted by many activated lymphocytes, macrophages, dendritic cells, epithelial cells, endothelial cells, or connective tissue. But these cytokines do not include the growth factor that act on epithelial and mesenchymal cells. There are many important cytokines in the process of inflammation. One group is interleukin. This interleukin is interleukin 1, 6, 8, 13, and 10, and these are important in inflammatory cell activation. Then there are various growth factors that are responsible in this inflammatory response. These are granulocyte, monocyte, colony stimulating factor, and monocyte colony stimulating factors, and they are responsible for macrophage activation and bactericidal activity. Also, they are important in function of natural killer cells or null cells and dendritic cell function. Then various chemokines, this classification of these chemokines we are going to see later, and they are involved in chemotaxis of the leukocyte and activation of the leukocytes. Interferons have got alpha, beta, and gamma group, and they have got antiviral activity. They are also implicated in leukocyte activation. Then there are pro-inflammatory cytokines. One important is tumor necrotic factor alpha. This is associated with fever, anorexia, shock, cytotoxicity, cytokine, and many other uh, activities. The cytokines of acute inflammation are TNF, interleukin 1, interleukin 6, chemokines, and interleukin 17. Tumor necrotic factor is secreted from macrophages, chiefly mast cells and T lymphocyte. It stimulates the expression of endothelial adhesion molecules and also causes the secretion of other cytokines. It is implicated in the systemic effects of the inflammation also. Interleukin 1 is secreted from macrophages, endothelial cells, and some of the epithelial cells. Most of its functions are similar to TNF, and it has got a greater role in development of fever. Interleukin 6 are secreted by the macrophages chiefly and some other cells, and they are responsible for systemic effect, especially the acute phase response in the acute inflammation. The source of chemokines are macrophages, endothelial cell, T lymphocytes, mast cells, and other cell type, and they are responsible for recruitment of leukocytes at the site of inflammation and migration of these cells in the normal tissue. Interleukin 17 is secreted by the T lymphocytes, and it is a subset of T lymphocyte, and it is responsible for recruitment of neutrophils and monocytes at the site of the inflammation. The cytokines of chronic inflammation are interleukin 12, gamma interferon, and interleukin 17. The interleukin 12 are secreted from dendritic cell and macrophages, and they increase the production of gamma interferon. The gamma interferon sources are T lymphocyte and natural killer cell. They activate macrophages and also increase their ability to kill microbe and tumor cells. Interleukin 17 is implicated in both the acute and chronic response, and in chronic inflammation they are responsible for recruitment of neutrophil and monocyte at the site of the inflammation. The tumor necrotic factor and interleukin 1 play their part in recruitment of the leukocyte by promoting the endothelial cell adhesion or immigration of leukocyte through the vessel wall. The TNF and interleukin are produced by activated macrophages and dendritic cell, while TNF is also produced by T lymphocytes and mast cell, while the interleukin 1 is secreted by some of the epithelial cells. The secretion of the TNF and interleukin 1 is stimulated by various microbial products, foreign bodies, immune complexes, physical injury, or many other causes that are responsible for this stimulation. Their production is induced by toll-like receptors recognition and some microbial sensors that are especially affiliated with the production of TNF, while the inflammasome activity, that is a multi-protein complex activity, it will result in interleukin production. The interleukin 1 and tumor necrotic factor are responsible for activation of the endothelial cell. There are many changes occur in these endothelium that are collectively called as endothelial activation. Then they are also responsible for activation of leukocyte and other cells and systemic acute phase response. So expression of adhesion molecule is increased by these TNF and interleukin 1. These adhesion molecules such as E and P selectins that are expressed on the endothelial cell, and they are responsible for uh, adhesion, and integrin ligand are also expressed more. Then production of various mediators such as cytokines, chemokines, growth factor, and eicosanoids, and the procoagulant activity of endothelium is increased. So these TNF and interleukin 1, they enhance the activity of these endothelial cells toward the inflammatory process. TNF augment responses of neutrophil to other stimuli, and they also stimulate the microbicidal activity of macrophages. Interleukin 1 activates fibroblast, hence there is collagen synthesis. The synovial and other mesenchymal cells also proliferate, and Th17 response is also stimulated by interleukin 1 that leads to the stimulation or induction of acute inflammation. Then the third one is systemic acute phase response. This is induced by tumor necrotic factor interleukin 6 and interleukin 1. Especially the interleukin 6 are responsible for fever and sepsis that occur in systemic acute phase response. The TNF or tumor necrotic factor are responsible for lipid and protein mobilization, and they are also responsible for suppression of the appetite that leads to the cachexia, a metabolic state in which there is anorexia and a loss of appetite in chronic inflammatory condition. Interleukin 1 is also associated in the systemic acute phase response, but its role is not well understood. So the TNF effect can be blocked by TNF antagonist, and it is part of treatment in rheumatoid arthritis, psoriasis, and inflammatory bowel disease, chronic conditions, and this use of TNF antagonist is associated with increased risk for mycobacterial tuberculosis infection because of decreased activity of TNF to kill the invading agent. Then role of interleukin 1 blocker is not well understood in systemic acute phase response. So this illustration is showing the central role of interleukin 1 and tumor necrotic factor alpha in the inflammation. The macrophages that are activated by the T cells through interferon gamma or gram-negative bacteria by the lipopolysaccharide, they are associated with endothelial cell leukocyte adhesion by tumor necrotic factor. They are responsible for recruitment of the leukocyte by interleukin 8. Interleukin 6 and interleukin 1 play their part in acute phase response, while is uh, there are many immune functions that are associated with this process through interleukin 1, 6 and 12. So these mediator TNF and interleukin 1, they exert their effect on endothelial cell. There is adhesion molecule expression, liberation of cytokines, eicosanoid, chemokines and oxygen radicals, while the effect on neutrophils are priming of the neutrophil and aggregation of the neutrophil. Then these are responsible for acute phase response that is characterized by fever, anorexia, hypotension, increased heart rate, corticosteroid and ACTH release. This picture is describing many local and systemic effect of these cytokines. Some are protective and some are pathologic. The local effects of these cytokines on blood vessels are interleukin 1 and TNF. They increase the expression of adhesion molecule on the endothelial cell, and the TNF is responsible for increased permeability of the vessel wall. The interleukin 1 and chemokines are also secreted by the activation by the local endothelial activation. Then the TNF and interleukin 1, they activate the leukocyte and also the macrophage and are responsible for release of interleukin 1, 6 and chemokines. Then this inflammation has got the effect of the cytokines during the inflammation has got some protective effect. TNF, interleukin 1 and interleukin 6 are associated with onset of fever. That is a protective mechanism that alarms the body of something that is going on wrong. Interleukin 1 and 6, if they act on the liver and are responsible for release of acute phase proteins, and interleukin 1, 6 and TNF, they act on bone marrow and they induce the production of the leukocyte. Then there are some pathologic effects. TNF decreases the cardiac output, and also it has got some thrombogenic effect. There is increased permeability of the vessel wall that results in edema. Tumor necrotic factor and interleukin 1 also affect on multiple tissue, especially in the skeletal tissue. They induce resistance to insulin. This table is summarizing the major cytokines and their source and function. The TNF alpha or tumor necrotic alpha source is macrophage, endothelial cell and leukocyte. It acts on endothelial cells and activate them. It also acts on neutrophil and primes these neutrophil and modulate the expression of adhesion molecules. And effect of TNF alpha are fever, aggregation of the neutrophils, there is muscle catabolism and shift in protein synthesis. Has also got hemodynamic effects. Interleukin 1 source are macrophages and endothelial cells. This interleukin 1 amplifies inflammation and activates the cells, endothelial cells. It also induces cytokine and reactive oxygen species release. Interleukin 1 is also associated with fever, acute phase response, recruitment of more leukocytes, and amplification of the inflammation. The source of interleukin 6 is primarily the macrophages, and they are implicated in acute phase protein production and immune regulation. They are also associated with fever, acute phase response, and immune modulation. Interleukin 8 from the macrophages and endothelial cells serves as chemoattractant for neutrophils, and it enhances the neutrophil recruitment to the site of inflammation. Interleukin 12 from macrophages stimulate natural killer cell and T cell to produce gamma interferon, and these cytokines then enhance immune response to intracellular pathogens. Gamma interferon that is produced by the NK cells, natural killer cells, and T lymphocyte are potent macrophage activator, and they enhance cytokine production. So they amplify the response of the macrophages and boost immune defense against intracellular pathogens. Now about the chemokines. These chemokines are a family of small proteins that act primarily as chemoattractant for a specific type of leukocyte. They are about 8 to 10 kilodalton in weight, and about 40 chemokines have been recognized with 20 receptors for these. They are classified on the basis of arrangement of cysteine residues in the protein, and the four major classes of these are CXC, and in which there is an amino acid between the first three conserved residues of cysteine. Then there is when there is no amino acid between these two cysteine molecules, they are termed as CC. And CC chemokines, when the first and third of the four conserved cysteine are lacking, this is C chemokine. And when there are three amino acids between the two cysteine, it is termed as CX3C. So this CXC chemokine has one amino acid residue that is separating the first two of the four conserved cysteine residue. Microbial products, cytokines, interleukin 1 and TNF are their inducers, and they are secreted by activated macrophages, endothelial cells and other cell types. Their effect is chemotaxis of neutrophils, and they have also shown limited activity on monocyte and eosinophils. The CC chemokines have first two conserved cysteine residue that are lying adjacent. They are attractant chemoattractant monocytic protein MCP1, eutaxin that selectively recruits eosinophil, macrophage inflammatory protein alpha 1, and rentase, the regulated and normal T cell expressed and secreted, are important member of these CC chemokines. Their effect is to attract monocyte, eosinophil, basophil and lymphocyte, and they are not a potent chemoattractor for neutrophils. Now the C chemokines, the most important and the specific chemokine is lymphotactin, that is specific for lymphocyte, and C chemokines, they lack the first and third of the four conserved cysteines. CX3C chemokines contain three amino acids between the first two cysteines, and the important members are fret alkyne, and this is a cell surface bound protein that is present in soluble form and induced on endothelial cell by inflammatory cytokines that promote a strong adhesion of monocytes and T cells. Now, what are their function? Basically, these function in acute inflammation, and these chemokines are induced by microbes and other stimuli, and then these increase the leukocyte affinity for integrin, and they also stimulate migration, that is chemotaxis, and they also maintain the tissue architecture by the presence of some homeostatic chemokines. The homeostatic chemokines organize various cell types in different anatomic region of the tissue. T and B lymphocytes in the spleen and lymph node is one of the example how these cells are arranged in a specific part, a specific area of the organ. This table is summarizing the chemokine, their classes, structure, main source, what are their function. So CXC chemokine has got one amino acid between the first two cysteine. The major source is macrophage and endothelial cell. The target cell is neutrophil. And activation of chemotaxis and activation and chemotaxis of neutrophil is their main function. An example is interleukin 8. The CC chemokines have got first two cysteine that are lying adjacent, and they are derived from various immune cells. The target cells are monocyte, eosinophil, basophil, and lymphocyte, and they attract these cells. Eutexin of the one of the member of this CC chemokine especially recruit eosinophil. Then C chemokines lack first and third cysteine, and are they are made from lymphoid tissue, and the target cells are lymphocyte. They selectively attract lymphocytes, and example is lymphotectin. When there are three amino acids between the cysteine present and the source of the CX3C chemokine is endothelial cells, they attract monocytes and T cell, and these chemokines, for example, fructalkaline, they exist as a membrane-bound and soluble forms. Membrane-bound form promotes adhesion, and the soluble form promotes chemoattraction. These receptors, they exert their effect by binding to specific receptors that are 7-pass transmembrane G protein coupled receptor. They are termed as CXCR or CCR, and these chemokine receptor such as CXCR4 and CCR5, they act as co-receptor for viral enveloped glycoprotein of human immunodeficiency virus. Sometimes in some patients the receptor for the virus is not present, so virus is not entered into the cell, and clinical manifestation of the virus, even with the presence of the virus, are not manifested. This video on cytokines and chemokines. If you have any question, write in the comment box. In the next video, we are going to discuss the plasma-derived mediators of inflammation.